Okay, so here's a nice phylogeny with pretty sound phylogenetic groups, pretty sound what we would consider monophyletic groups by most. But these are big groups, right? These are big old groups. And so you can see a bunch of names up here. So this is not just vertebrates. Vertebrates and chordates are right over here. So this is the rest of animals. This is a big, huge, generic phylogeny of all multicellular animals. So you got some names up here. Let's go through the names. So these groups, let's just go through them real quickly again. So metazoa means tissue, right? So tissued one or one with tissues. And you can see it kind of goes across here, right? Where you have cells that are actually aggregated together and are working together to do something, right? That's what a tissue is. When you go below that name there, metazoa, you see eumetazoa and parazoa. And I don't even think I even mentioned parazoa last time. So the difference between these two groups, this means true tissues. This means without tissues, right? That's what para means. And so if you look down below para, what is our outgroup here in this large phylogeny of animals are peripherans, all right, or what are called sponges. And sponges don't have any true tissues. Now, they have aggregates of cells and obviously some glue that holds them together, but they don't have true developmental germ layers or tissues like we talked about with the rest of animals. So they're the only group without true tissues or true germ layers during development, like eumetazoans are. Now, when you look at eumetazoans, the rest of animals, right, we have radiata and bilateraria. Our radiata are obviously just cnidarians and ctenophores, and they have radial symmetry, which means they have a central point, and then the rest of their body radiates from that central point, like sun rays coming off a circle, right? So they're circular usually or oval shaped. They have a medusa. The other thing that I don't think I really made clear is that radiata is also diploblastic, right? It has two germ layers. And the two germ layers that develop are ectoderm and endoderm, right? So these guys only have two germ layers. The rest of animals have three germ layers and are called triploblastic. And they include what are called the coelomates. So you have acoelomates, pseudocoelomates, and coelomates here, right? Or what they call true coelomates or eucoelomates. And all of these have all three germ layers as you go from flatworms, which is Platyhelminthes, all the way across to chordates. The difference is in how the coelom forms. Obviously, an acoelomate, A means without, doesn't have a body cavity. That's what coelom means, right? Is body cavity. So they're solid all the way through. They're also flat like my hand, like somebody just sandwiched them down or flattened them. And that's what platy means. Platy means flat. Henthes means worm, so flatworms, like planarians. The rest of the organisms here, from Nematoda across, they have a body cavity. The difference between a pseudocoelomate and a coelomate is whether the body cavity is completely lined with mesoderm or not. That is, you can see mesoderm, lines the entire cavity. True coelomates have mesoderm that lines the entire cavity. Pseudocoelomates only have partial covering of their body cavity with mesoderm. And so their coelom's not true because it's not lined nicely with mesoderm. That's the difference between the two. But all three of these, acoelomates, pseudocoelomates, and coelomates, are triploblastic or have ectoderm, endoderm, and mesoderm, the three germ layers that we talked about earlier. And then notice protostomians and deuterostomians are, remember, two of our, what we're going to call infrakingdoms. Remember that? And we talked a lot about the differences between these guys, and that proto means first, stomia means mouth, deutero means second, mouth. And remember, during gastrulation, the blastopore forms, right? That little hole that is going to empty into the digestive tract. In protostomes, that hole becomes the mouth. That's why it's called first mouth. In deuterostomes, the blastopore is fated to be the anus, which is our second opening, right? First opening, second opening. That's why it's called second mouth. But we're going to really focus now on the triploblastic guys. All right, so the guys that are true coelomates from mollusks to chordates. And we'll compare those in the next slide. The other thing I like to throw in before we jump into development is, species, the number of species or how diverse groups are. And I always like to throw this in there because everybody thinks vertebrates are so successful, right? Because that's what you see, and they're big and they take up lots of space. But a lot of biologists agree that it's the diversity and number of species that actually determine whether you're successful or not, right? And so look at the major groups here. So these are some major groups, kind of at the order and class level, and you can see arthropods. Arthropoda here is number one. It's got a number of species that are above 1 million, right? And then you got to go all the way down here to number 4 to get the chordates. So the invertebrates are very adaptive to where they live and how they do things. They're very successful. Chordates are very successful too. We're just bigger and take up more space and reproduce a bit slower in most cases than the invertebrates do. And then these are some of the other groups that we just mentioned. And then there's also some phyla we didn't talk about at all, which are really small. Because this isn't zoology, right? This is vertebrate anatomy. So we want to get into the vertebrates. So deuterostomes, what's the difference between deuterostomes and protostomes? Or what's the difference between chordates and echinoderms and the rest of triploblastic phyla? So these two development types, they're found in invertebrates. Both of them are. So invertebrates run from mollusks to the echinoderms on our chart, okay? And the echinoderms, Echinodermata, is a deuterostome. All the rest of the invertebrates are protostomes, all right, and belong to protostomia. So what's the difference between these if we're just focused on the triploblastic ones, right? The triploblastic ones, the differences are in early development. That's this first box here. In early development, you undergo a process that's called cleavage. All right, and what cleavage is fast mitosis. All right, so when sperm and egg meets, they form a new cell, right? And that new cell we call a fertilized egg, right? Or we can start calling it an embryo at that point, right? That single cell is going to immediately divide really quickly. And, it just depends on the organism. It could be 24 hours of really fast division. It could be 72 hours of division, whatever it is. But no matter what type of triploblast you talk about, the first thing that happens in development is immediate cleavage to divide our cytoplasm up into smaller units that we can use. And the way they cleave is unique to protostomes versus deuterostomes. So the best way to describe it is if you were to look down on a protostome, the cells are kind of off center. And so they look like a spiraling staircase as they're added to the top of the embryo. So they call it spiral cleavage as they go up. Because the plane, notice the plane of cleavage here, which is the middle of the cell where you divide the cells in half, are offset. See that? There's one here in the middle of this cell, and then there's one there. And so it looks like they're spiraling towards the middle. So it's called spiral cleavage. The other type is called radial cleavage, where you radiate out from a central point. And that's because all the cleavage furrows or all the cleavage planes line up real nicely on top of one another. And so it looks like you're radiating out from a central point. And so they call it radial cleavage. The other term here, determinate versus indeterminate. And this is up through it depends on who you read, but up through the 6 to 8 cell stage. Once you get to the 8 cell stage, all protostomes, these 8 cells are determined to be something. They can't be told to be something else. So if you're a cell that's going to end up being part of the arm or leg or appendage of the organism, that's your fate. You're going to be appendage. No matter what I try to do to the cell to make it do something else, it will not. Whereas a deuterostome, up through the 8-cell stage, these cells can become anything. You can tell them to become anything. So what that means is, let's say we have a deuterostome that splits into 4 cells. If we break that embryo up, those 4 cells will become 4 embryos. It's because they're not determined yet. They're called indeterminate. Some of their genes haven't been turned off yet, and are still available to make whole embryos. So it's called totipotent is the name. Toti, T-O-T-I. Potent means I can be anything. Total embryo. And so that's a little different. Now, past the 8-cell stage, everybody's determined to be something as far as what they're going to be in the adult organism. Now, what happens with development is you divide and divide and divide and divide. Here's an example of a embryo that is done dividing, done doing cleavage, termination of cleavage. And you can say what's really cool is the original single cell embryo that we started with, the size after cleavage is no different. It's almost the same exact size as the sperm and egg and when they met, or it's the same size as the original egg. All we've done is divided the cytoplasm up into usable units, right? Smaller units so that the cell can do well. So you can start counting cells here, right? And so some animals have tens of thousands of cells after cleavage. Some of them have 100. I mean, it just depends on the organism and how fast cleavage is. But the end here, the end of the cleavage stage is called the blastula, right? So the blastula is the name of the end product of cleavage. And that blastula is hollow. It's a hollow ball of cells. So once we're done with cleavage, we're sitting here with a hollow ball of cells, right? The next stage is called gastrulation. Sometimes cleavage is called blastulation because you're building a blastula, right? And so gastrulation is the next stage. And so what happens? In triploblasts, one end of the embryo is going to buckle inward. So you can see right here. So here's my embryo. I'm looking down on a microscope, and I've focused on the body. I shouldn't say body. It is going to become the body cavity. But the lumen or space of my blastula, I'm looking in. And you can see here, gastrulation has already started. So notice the underside of the embryo has buckled, and the cells on the outside are being pulled in as a tissue. You see that? It's forming a new pore and cavity. Right? This new cavity that's being pulled up has a name. It's called the archenteron in development. And it's going to continue to elongate until it pierces the other side. Okay? So you're pulling the tissues and cells from the outside inward. You see that? Into the middle of the hollow ball of cells. And this archenteron is going to become the digestive tract. Right? That's going to be the hollow tube we call the intestines and the stomach and whatnot. And so you can, this pore here called the blastopore has 2 different fates in our 2 groups. In protostomes, it's the mouth, right? It's fated to be the mouth, the opening of the mouth. In deuterostomes, it's the anus. So it's opposite. So when this tube finishes to the other side, in a protostome, it becomes the anus, and in a deuterostome, it becomes the mouth. Right? So it's just, slight differences in development. The tissue that lines the archenteron is called endoderm, right? Like I mentioned before. All right, so endoderm. As you can see here, it's labeled in yellow. And the outside is going to be called ectoderm. Remember, those were the 2 layers that formed in a diploblast, right? There's a 3rd layer that forms about mid-gastrulation. We get the tube about midway up. There are going to be some cells in the endoderm that change shape, release from their basement membranes, and move into the body cavity. And you can see these guys. See them coming off of the body cavity here? And they will become and start dividing and become the third tissue type, which is mesoderm. And mesoderm will fill this body cavity up. All right, and so it's the middle layer. So you got ecto means outer, endo means inner, and meso means middle, right? The middle layer. Now, there are two ways that mesoderm forms. It can form like this, where it is kind of falling off. These cells move and fall off and into the body cavity and then divide, like you can see where number 3 is, and start dividing to fill in the space. All right, that is called schizocoelom, all right, or schizocoelius, which means out within or outside in the coelom, outside of the endoderm. Or it can be called enterocoelus, which means within the endoderm. And this is the difference between protostomes and deuterostomes, right? Protostomes, the cells fall off into the body cavity and then start dividing and form their own little ball of cells. Whereas mesoderm in the deuterostome first forms as little Mickey Mouse ears, at least that's what I call them. They look like little Mickey Mouse ears on top. And so they divide and form these little evaginations that eventually pinch off as balls of cells and then fill in the body cavity. Now, ultimately, what the mesoderm is going to do is divide and spread out and line the entire cavity. So they're going to line the underside of the ectoderm, and they're going to line on top of the endoderm. And they form a true body coelom. A true body coelom is lined completely with mesoderm, right? And that forms only when you get mesoderm, right? Forming as a third tissue. So this forms during gastrulation in triploblastic bilateral oriens, right? Or what we call left and right half organisms. And you can see this is the line, the deuterostome line that the chordates follow. So you're a deuterostome. We're mammals, we're vertebrates, we're a chordate. The other, I just threw this term in here. The other term that you will see a lot here when we start talking about some anatomy and architecture is soma, which means body. And it's an old school term for body. If you've had any nerve anatomy, the nerve body is called the soma, right? Viscera means body organs. Viscera means body organs, and we'll all use that term a lot. The other thing that I want you to notice here is that with protostomes, you often form a type of skeleton, right? Something that supports the body. And the skeleton in protostomes is ectodermal, which means it forms in the skin or the outer layer. The ectoderm gives rise to the skeleton. Whereas in deuterostomes, particularly in vertebrates, the skeleton is mesodermal in derivative. It comes from mesoderm. Bone, muscle, everything that you can think is kind of between the organs and the skin is typically at least part mesodermal, right? Which makes sense because of where it's located. Okay, we're going to start talking about groups of vertebrates here, or chordates, I should say. We'll move into the vertebrate groups and try to make sense of the taxonomy here, which can be really confusing because nobody agrees on it. But back when I learned about these guys a long time ago, we called them protochordates, or proto means first, or primitive chordate. And these are the 3 groups that I have circled here. So if you remember here, we have some bilateral orien ancestor that had a right and left side, right, that were mirror images of each other. And we can break those, what we call true coelomates, down into protostomes and deuterostomes. And the protostomes are all the invertebrates, I'll show you a picture of those, a better phylogeny here in a second, like, arthropods, insects, worms, mollusks, octopi, those kinds of things. And then you have your deuterostomes over here. And in this really old phylogeny, you have 2 phyla here that are within the deuterostomes. One's called echinodermata, and the other one's called chordata, right? Or chordata. Some people say chordata. And they share all the characteristics or suite of characters that we use for deuterostomes and protostomes. And I don't know if I told you the origin of the names. The origin of the names always help me a little bit, when I'm looking at them. So proto means first, like I just said. So stomi or stomia, the Greek is mouth. So first mouth, that's protostomes. Deuterostomes means second mouth. So what does that mean? First mouth means that the blastopore becomes the mouth, which is what happens in protostomes, the first hole, right? Where deuterostomes, the blastopore becomes the second mouth, or what they call the anus. That's where the name protostome and deuterostome comes from. But the deuterostomes are these two large groups, and we're going to talk about phylum Chordata here and start moving into that. So when you look at this phylogeny here, it gives you a number of characteristics that are found in each of the branching points. And notice our root here, or our outgroup, is our sister taxon. These two are considered sister taxon because they share the most common recent ancestor, echinoderms. And this term, echinodermata, means spiny or spiked skin or hard skin. I've seen it translated like that also. And that includes, think of starfish. I don't know if you've ever held a starfish, but they're kind of a spiny, hard skin. So you can see they root our tree, and then these are what are considered our Chordata phylum here. And so notice the first three groups, hemi, euro, and cephalo, all end in chordate. And those are our three, what we call primitive groups, our protostomes. Uh-oh.
Sorry.
Oh, you got your watch pointer? Oh, okay. So these are, which mean primitive or first groups. And so this used to be considered a phylogenetically sound taxon a long time ago, like in the 70s. But now it's considered polyphyletic because hemichordates, as you're going to see here in a second, are more closely related to echinoderms than they are euro and cephalochordates. So that would be an example of polyphyletic, right? Two different origins. But we still teach them together. So let's start diving into these. So we'll talk about the hemichordates first. So what makes a chordate, right? Compared to all the other organisms out there, all the other animals. And it's these 5 unique characters that are found in all chordates. So we would call these plesiomorphies for chordates, right? Or apomorphies for chordates if you do compare them to other animals, right? But these characteristics, the suite of characteristics here, are found in all chordates, which includes you. Now, the one major thing about the characteristics is they have to arise sometimes during your life cycle. They don't have to be present all the time. Heck, they can be present in utero in us, and you never see them in the baby or the adult. But they are there during development. So here are the 5 characters. They're called the notochord, the dorsal hollow nerve cord, or what we call the spinal cord, right? Pharyngeal or stigmatal, or stigmata, slits. These are not gills. They're what we would consider true gills. Sometimes in textbooks you see them called gill slits, or especially human textbooks during development, they'll call them gill slits, but they're really not. They're pharyngeal slits. Postanal tail, and a thyroid gland, or another name for the primitive thyroid gland is an endostyle. You can almost use them interchangeably in my book, but those are the 5 characters. And so if you look at a chordate, you should be able to find those characters sometime during the life history. So this picture here, this is the front half, or the cranial half of one of these, what we call primitive chordates, these protochordates. This is called Amphioxus. I'll bring some up. I have some down in, I think I have some in my lab downstairs. They're about this big. They're a couple, I can see them, I've seen them about that big, about 8 inches, but they're typically, 2 to 3 inches long. And so this is one stained under the microscope. You'll be able to look at some like juvenile Amphioxus on your microscope slides next week. We'll start looking at them. But they show all the characters. So let's go through these characters. So the notochord is the first one. So notochord is a gelatinous rod, basically what the name means, or gelatinous thread. I like rod. And so the notochord runs along the dorsal surface. So this would be ventral down here, ventral surface or belly. This would be dorsal surface or back. So this long, dark cylinder running right here is the notochord. Notice it runs all the way into this little rostrum or this little appendage that sticks off the front of the head. This would be the cranial end or the head of Amphioxus. And so this notochord developed in fossils around 500 to 480 million years ago. And what this rod does is allow for better movement forward. And it basically gives the body some structure so that you can use muscles to bend it. And since it's not a collapsible rod, it allows the animal to propel itself forward or to form a more effective tail and then swimming motion. The notochord eventually will be replaced in vertebrates by the vertebral column, right, by bone. You still see remnants of the notochord even in us. Our notochord is found in our intervertebral discs. The discs, the cartilaginous discs between vertebrae. In the middle of that, when you take in a human A&P course, they call it the nucleus pulposus. I'll show you a picture of it in a second. That is the remnant of our notochord. All the rest of it has been replaced by our vertebral column. So the notochord is found in all chordates. So to be a chordate, you have to have that long rod on your back. Of course, ours is replaced, right, by the vertebral column. Dorsal hollow nerve cord sits on top of that. It's the nerve cord that runs down the length of the body. And it's this lighter orange structure on top here. And you can see these little dashed lines. Those are pigmented cells. But this long kind of light orange rod that runs or sits on top of the notochord is the dorsal hollow nerve cord. To give you a comparison, most invertebrates have a ventral nerve cord. Right? It's on the belly, and that nerve cord is solid. It's not hollow like ours. Hollow meaning this is really a tube that's filled with fluid. Right? And the fluid is called cerebrospinal fluid, or CSF fluid, right? Cerebrospinal fluid, which is a fluid that bathes the inner chambers of the central nervous system in vertebrates. Pharyngeal or stigmatal slits. So the pharynx means throat. So pharynx means throat. All chordates have slits in their pharynx. So here's the big tubular pharynx right down here. It's kind of in the middle, sandwiched in the middle. And you can see it is not solid. It has slits in it, right, with bars. We developed these during early development. They obviously then migrate and become other things in our body because our pharynx is not slitted, right? It's solid. But when the pharyngeal slits or bars form in humans, they become other structures, like the hyoid bone, the little ear bones that are found in the inner ear. Your lower jaw is a pharyngeal slit. So they migrate and form different structures in the head and neck. Postanal tail. All chordates have a tail past the anus sometime during their development. Ours falls off, right? Or fall off. I shouldn't say that. It doesn't really fall off. It's reabsorbed during our development. Do you know every once in a while a human's born with a tail? And because it wasn't reabsorbed or underwent programmed cell death, so they have to remove it. But all tailless vertebrates had a tail during development. And then the last thing is an endostyle or a thyroid gland, which sits around the pharynx, somewhere near the pharynx. Ours sits right on top of our larynx. All right, that's where our thyroid gland is. But in, for example, Amphioxus, you see this dark bar on the bottom or the ventral surface of the pharynx? That is the endostyle, all right, or the primitive thyroid gland, or what we call the ancestral thyroid gland. And so it forms the floor, if you will, of their pharynx. And it has two functions in these protochordates. It produces mucus, lots of mucus. The second job of it is to concentrate iodine. Right? Iodine is one of the elements needed to make thyroid hormone. And so it can concentrate iodine and make iodine later, or I should say make thyroid hormone later in the life cycle. So in order to make thyroid hormone, all thyroid glands have to be able to sequester or concentrate iodine. So any iodine that comes into your body, almost 99% of it, ends up in the thyroid. The other 0.5 to 0.1% of it ends up in bone tissue. So, that iodine is used to make thyroid hormone. Now, this is a cross section through this organism. So if I cut straight down like this and then pull the front half off and look in, right? That's what a cross section is. This is a sagittal section, right? Or a whole mount. And this is a cross section through actually the intestinal region. So the cross section is actually down here. It's a little further caudal, right? Or towards the tail. And so you can see here, this is the notochord. Look how big it is. It is a large structure in this organism. So there's the notochord in cross section. Here's the dorsal hollow nerve cord, right? This is the intestine. These are gonads. They're actually testes. But you can see at least two of those features right there, right? Now, if we section through about, I would say right in this area right here, if you follow my pointer here, if I section straight down there and take the cranial end off and look in, then I'll see part of the intestine and part of the pharynx, right? And that's what you see here. So here's part of the pharynx. Here's part of the intestine, right, in cross section. These are, again, gonads. These are ovaries. These big round cells are oocytes. Here's a blow up of that area, right around here. So here is the intestine in cross section. Guess what? It has a simple columnar epithelium, just like ours. These are ovaries, one ovary, two ovaries. This is basically the ventral side, or the ventral compartment of the pharynx. And what you'll notice about the pharynx is it's got spaces. You see that? Between the bars. And so it's got slits, and water leaks through those slits. And we'll talk about how the pharynx works with this particular organism when we get to it. So there's your pharyngeal bars, your pharyngeal slits. Again, notochord, dorsal hollow nerve cord. There's muscle, notice on either side. This is skeletal muscle tissue on either side of the notochord, so that you can do this. You can bend it like that and like that, right? And so if my arm's the notochord and you're bending it, then you can do this to the tail, right? And then you can swim really quickly forward. Wonderful invention of evolution. The ability to move forward quickly to be a potent predator or to escape predation. These guys are good swimmers because of the notochord. So those are the 5 unique characters. We should be able to see them in all of the chordates that we look at. Now here's a picture of what you will see when you actually look at your slides. So I got whole mounts of these guys, and I got cross sections of them, which we'll start next week. And this is on I think it's on 10x, and obviously blown up on the board. That shows the ventral surface of a cross section of just the pharynx. All right, so this is near the belly, through the pharynx, and this is the pharynx in the middle here. So you can see it. These structures labeled B are bars, pharyngeal bars, and then C are the slits. Now, if this is the floor of the pharynx, then this is the endostyle in cross section. It looks like a little horseshoe. So what I always think it looks like, or a little U, right? And so this structure, what it does is it produces mucus. So it's going to produce mucus, and that mucus is going to kind of flood into the lumen here of the pharynx, and it's going to cover these bars. And it moves because the bars, the epithelia that line the bars, are ciliated, so they beat, right? And they can push the mucus up and cover the bars in mucus. And so imagine this. If I bring water through the pharynx, I suck it in, right? And allow the water to escape through the slits, then I can collect food from the water by it sticking to the mucus, right? The cilia beat up and push the mucus towards the dorsal end or the dorsal side of the pharynx and then collect all that mucus-laden food or mucus with lots of food in it and then push it into the gut. And that's kind of how they feed. They're filter feeders. Now, again, I leave this figure on here. I didn't make this figure. I stole it off of a website a long time ago. I can't remember the San Diego State, maybe University, and it's labeled wrong. So this is the problem with the internet. So they label these as ovaries over here, and being a reproductive biologist, that's a testis. So they label things wrong. I mean, it happens, right? Things get labeled wrong. I've labeled things wrong too. Don't get me wrong. I make lots of mistakes. But if you see something and you're like, that's not right, or Gribbins, that's wrong, what you label, that's probably wrong. I probably labeled it wrong, right? So call me out or call people out on it. The rest of it's right though. Now these are old. These are old names. Notice this is how this shows the age, because they're showing phylum Chordata and then subphylum Cephalochordata, and we're going to change that up a little bit as we go through here. So you'll see some changes in taxonomic hierarchy. All right, so what makes an endostyle, which is this, right? A thyroid gland. Well, interestingly, the endostyle really in what we're going to call our really, really ancestral chordates is really a mucus-producing structure. It produces lots of mucus. And it uses that mucus is used to help it, capture food. It does concentrate, always concentrates iodine, and it will make a little bit of thyroid hormone, particularly when metamorphosis happens between the larva and the adult. Because thyroid hormone controls metamorphosis in vertebrates, okay, or all chordates, really. And so you'll get this burst of thyroid hormone, which tells the larva to become the adult, right? So, for example, when a tadpole becomes an adult frog, there's a big burst of thyroid hormone, right, that causes that. Same thing in all chordates. But really, that's a really short time frame within Amphioxus, for example, the organism that's more ancestral than a lamprey, for example. And so their endostyle produces thyroid hormone just for a very short time during the life cycle, and it's producing mucus the rest of the time. So this is that's kind of what an endostyle looks like in Amphioxus, right, or in a cephalochordate. Now, lampreys are a much more derived chordate. They're actually a vertebrate, right, which is a more derived chordate. Interestingly, in the larva of the lamprey, so this is a lamprey larva, man, it looks a lot like Amphioxus, doesn't it, morphologically? I mean, you might look at it as a novice and be like, that is Amphioxus. I mean, it looks just like Amphioxus. But there's a bunch of differences in the head. For example, there's an eye and there's ears, ear vesicles. There's a lot more derived structures in the head. Whereas if you look at Amphioxus, there's no eye, right? There's no ear. You'll see that when you look at this guy up here. So he's got an eye there with a little dot in it, and then there's an ear vesicle behind. The mouth's a little more complicated, but the rest of this larva looks a lot like Amphioxus. Now, early in the development of, or growth, I should say, of the lamprey larva, if you look at the floor of the pharynx, and here's the pharynx, right? Here are your bars and your slits. It has a nice endostyle. The endostyle looks just like Amphioxus's endostyle. You look back another 3, 4, 5 days later, and this is what it looks like now in the same place. So the endostyle has been replaced with these big, huge clumps of cells that have a good circulatory component to them, some blood vessels that run through it. And now this thing is not producing mucus like this structure. Its whole job is to produce thyroid hormone. Okay? Now there's still a remnant endostyle on top. This is the floor of the pharynx, but notice the endostyle is now developed into a completely separate organ that runs the length of, or almost the whole length of the pharynx. It's this big dark structure right here, and it's called the subpharyngeal gland because this gland they call the thyroid gland. sometimes the thyroid gland is called the subpharyngeal gland because it is on the belly side of the pharynx. So it's pretty cool. You see an endostyle in a lamprey that then develops directly into a thyroid gland, which it means it's a really good example developmentally that the endostyle is actually a homology, right? It's actually an apomorphy, or I should say a plesiomorphy of vertebrates. So they inherit that from their chordate ancestry. So that's pretty interesting that you can see that, whereas in the Amphioxus, it just stays the endostyle. So this is directly out of a paper that was done in 2005. So pulled those images out of that paper because they looked at the development of the thyroid gland in the lamprey. All right, the notochord. So the notochord is also, you can trace it during development very nicely. So you can see it in a really complicated derived vertebrate like birds. Birds are a great example to use because, like if you look at a chicken, for example, chicks, which they use a lot, the animal part of the egg is actually a disc. It's thin and it floats on top of the yolk, right? And it develops into the chick on top of that yolk. And so you can see on the dorsal side of that animal disc, during the right time in development, which is after gastrulation and start of organogenesis, the development of organs, you'll start to get a thickening of the dorsal side of the embryo. And this is what's called a scanning electron micrograph. And what I've done is I've taken the egg, I fixed it with a fixative and made it really hard, and then I cut it cross-sectionally so that you can just see the rest of the animal is extending backwards here. I've taken off, again, the cranial end of the embryo so you can look in. And this here is ectoderm. All right, you can see the layer of ectoderm, right? These a single layer of cells, the outer layer or germ layer of the embryo. And notice it's thickening and forming this valley, if you will, right there. And this is called the neural plate. And these are called neural grooves. And notice the grooves, the cells are dividing down here towards the base and moving and growing as they move up, and they're forming these grooves. And if the growth continues, what's going to happen here are these two edges of the grooves are going to meet, aren't they? And form a complete tube that's going to zip closed. It's pretty cool. Typically it zips from caudal to cranial or from tail to head. And it will zip close and form a hollow tube along the back of the organism, which is going to become the spinal cord, or the dorsal hollow spinal cord, right? Or dorsal hollow nerve cord. Interestingly, in vertebrates like us, what causes the differentiation of that ectoderm to neural plate is the notochord. So the notochord actually develops where the vertebral column is going to develop, right, on our backs. And then what it does is it tells the ectoderm above it, or dorsally to it, to become nerve tissue, right? And it sends little chemical signals up into the dorsal ectoderm and says, hey, become nerve tissue. And that's why the neural plate forms. And I'm just gonna, I put a new picture in that yesterday, and I can update the PowerPoint for you. That shows this, right? So this is early. This is a little bit earlier in the chick. And then I went out and found a paper, and these are two different images. This one shows you now that my edges have come together, right? And they're touching. And here is the notochord. It's kind of sandwiched. And here's my ectoderm. If you follow the ectoderm up, this is going to become skin. But notice it then dives down, and it's pinching off. And this is the part of that ectoderm that became the spinal cord in cross-section. Here's the notochord. It runs underneath or ventrally to that hollow nerve cord. Here's the nerve cord later on in development, still hollow. Here's the notochord. You can see it much better. So this is why the nerve cord sits on top of the notochord, right? Even in us, in vertebrates. These are the somites. Remember I showed you those yesterday, or was it yesterday? Was that yesterday or the day before? I can't remember. These are the groups of cells that migrate to the area next to or laterally to the spinal cord, and then that's a staging site. And then they're given instructions through chemical signals, and then they migrate out to become different types of cells. So melanocytes, you guys know melanocytes? They give your skin pigmentation. They arose in the somites. And then they migrated to the skin to give skin color. So I'll update your PowerPoint so you have those images, but they show you how important the notochord is. It's not only the staging or workbench for building the vertebral column, but it's what causes nervous tissue to form in most vertebrates. Now I'm going to back up to this picture. So the notochord, what the notochord looks like, this is a drawing of the notochord in cross-section from the textbook that you can get optionally for this course. And that shows you what a typical notochord looks like, even in us, during our development. Now, interestingly, if you look at the middle of an intervertebral disc, which is a cartilaginous disc that sits between the two vertebrae, so that way you have cartilage between bones so that our vertebral column is bendable, right? So you can bend your back and whatnot. These discs are responsible for movement. They allow for absorbing of shock. And in the middle of that disc is a kind of gelatinous, jelly-like structure that's called the nucleus pulposus, which looks just like this picture. This is an image through an intervertebral disc showing cartilage on top, cartilage on bottom, and this is what's sandwiched in the middle. These large circular structures that are gelatinous or fluid-filled, and they look just like the middle of the notochord, don't they? There's what's left over of our notochord after the vertebral column has formed. Now, the notochord itself is filled with fluid. These are filled with water, these spaces that are then lined with cells. They're vacuolar spaces within the notochordal cells. They're filled with water and some other solutes. But because they're filled with water, and you can continue to fill those spaces with water within those cells, that this is a hydrostatic structure, which means water is what gives it its structure. Now, the reason for that is because if you notice on the outside, there's really thick layers of connective tissue that are wrapped tightly around this core of notochordal cells and their fluid-filled vacuoles. So it's like taking really thick saran wrap and wrapping the notochord really tightly all the way along its length. And this connective tissue sheath is very heavy in collagen. So notice collagenous. Collagen's very strong, very tough, doesn't stretch. So this really thick connective tissue sheath is non-stretchable, which means it's not like a balloon. If I continue to fill it with water, it's not going to expand. It's going to stay at the same shape. Therefore, the more water I add to it, the more pressure I'm going to add, right? Because the water is going to push against the walls. The harder it is, the harder it is to bend, right? So there's this happy medium, enough water so that I got a nice rod, but I can move it. Or if I fill it with water, I can make it really hard and now it won't bend, right, as easily because that water is pushing against the sheath and preventing it from bending or making it more stout, more rigorous, however you want to think about it. And so that's kind of how it works. That's what a hydrostatic structure is, using water pressure to give you shape and rigidness. Does that make sense? Now the notochord comes from mesoderm. Remember, mesoderm is the middle layer of the developing embryo, right? The nervous tissue came from ectoderm, right, which is the outer layer. Now this last picture here shows you a arthropod versus a chordate. And what I want you to notice is their nerve cord is found on the ventral side of the body under the digestive tract, where our nerve cord is found dorsally on top of the notochord or within the vertebral column if you're a vertebrate. Both of them come from ectoderm though. So interestingly, and the other major difference here is your invertebrates have a solid nerve cord, where our nerve cord is hollow, right? It has what's called a neuroceal. So seal means body cavity, neuro means nervous. So neuroceal means a hollow tube or a hollow space within the tube that's filled with cerebrospinal fluid. Okay, questions on nerve cords? Okay, your favorite part, taxonomy, right? So this on top is how I learned it back in the 90s. This is the new scheme. Well, I mean, at least since I've looked, this is 2018-ish. I don't think anything else has really come out. There's this huge study that was done that took all the morphology that we knew and then compared it to a molecular data set where they sequenced a couple of genes and compared them to one another to build these phylogenetic trees. They put the morphological tree over top of the DNA tree, and they built this beautiful phylogeny. And then they had to fix the taxonomy, right, on top. So notice back in the day, these names here were just kind of rubbish. They were just kind of names we used to group things and weren't really phylogenetically sound. So you have kingdom here, and then phylum, and then subphylum, right? Remember kingdom, phylum, class, order. Now we gave these guys names because they ended up being phylogenetically sound once we did some molecular biology on them. And so we had to give them different names. So notice our kingdom's still on Amalia. Now we're going to add two smaller groupings to our kingdom name. So we got kingdom on Amalia, and then we got subkingdom bilateraria, right? Our bilateral organisms. And then up here, these would be radial organisms, right? They call them non-bilaterans here. So that's our subkingdom. And then within that subkingdom, there are two major infrakingdoms, protostomia and deuterostomia, right? Our protostomes are almost all of our invertebrates, and our deuterostomes are two groups. Now, remember, in that old phylogeny I showed you, notice up here that the hemichordates are still grouped with echinoderms. We used to group them together with the chordates, right, and call them protochordates. But notice the echinoderms and the hemichordata, they make up a new group called ambulocaria, all right? So ambulo-car means to walk on something, to walk on the ground is the literal Greek translation of it. The ambulocarum is the starfish arm with its tubed feet and how it ambers along the floor of the seafloor. So that's where this name comes from. And there are two groups. There's hemichordates and echinodermata. So they make up two sister taxon that belong to this monophyletic group. And then notice you have chordates down here, and there are three groups to the chordates: cephalochordata, urochordata, and then vertebrata. I like the term craniate. So I use this term, craniata, instead of vertebrata. And I'll tell you why when we get to them. Now, notice there are these four major supraphyla because they each are made up of multiple phyla. So this term here, ecdyzoa. So this term, if you look at the Greek breakdown of it, ekdys means to shed. Zoa means animal. So it's an animal that sheds its skin. And so these three groups are, the first two are worms. There's nematodes and Priapulida, which basically, if you want to know what it translates into penis, believe it or not, because these worms look like a phallus. Yeah, they really do. They're called penile worms. Nematodes are roundworms. Nemata means thread, and toda means form, so they are roundworms. And then arthropods, arthro means jointed, and poda means feet, so jointed appendages, where that name comes from. So these are insects, these are roundworms, these are penis worms, okay, on top. You can tell your mom that your professor said penis today in lecture. Anyway, that's ecdysoans. The next group here are called Lophotrochozoa. Right? It's a big name. So lopho means crest or tuft. I like tuft. Trocho means a skirt, like a skirt of ropes or a skirt of cilia. It's basically in terms of the cilia that wrap around it. And then zoa means animal. So these guys have basically crested, ciliated bodies and are bilateral organisms. And so they have a head that's kind of got a tuft of tentacles. That's the lopho part of it. It's called a lophophore that captures food. And then the trocha is the cilia that wrap around its body and they spin, right? So that moves the whole organism. And zoa means animal. And so this includes flatworms, platyhelminthes. Platy means flat, heminthes means worms. Mollusca, mollusks, which means shelled one, which includes things like snails and clams and shellfish. Annelids means segmented worms. So those are like earthworms and some of their relatives. That's the Lophotrochozoans. Those two, Ecdysoa and the Lophotrochozoans, make up Protostomia. Deuterostomes. We've already kind of started talking about these. So Ambulacraria echinodermata are starfish and sea urchins, two major groups. And echinoderm means hard-skinned or spiny skin, is where the name comes from. And then hemichordate means half a chordate. And the name hemichordate works because these guys have some of the chordate characters, but not all 5. So they're going to be a great outgroup, aren't they? Right? Because they have some of the characters, but not all of them. All right, your chordates all develop a notochord and have all 5 of those chordate characters. Those would be symplesiomorphes for chordata. And we have 3 major phyla: Cephalochordata, Urochordata, and Vertebrata. Now, when I was young, what we had it like this. This is called a trichromy, because we didn't know how these 3 were related to each other. Now we do because of the DNA. Cephalochordates are our ancestral group, and then these are our 2 sister taxons. So urochordates are more closely related to vertebrates or craniates than they are to cephalochordates, and cephalochordates are more related to these guys over here, believe it or not, than these 2 to each other, right, based on the DNA. Now, the other 2 pictures I have here are pretty interesting. So this is a Tornarian larva. This is a diplural larva. So this name, diplura, means 2-sided. Little 2 sides is what it means, literally the Greek. And so it's bilateral. You cut it in half, there's bilateral mirror images. Tornaria means to twirl, because these guys twirl when they swim. That's where the name comes from. But this one here, so the diplura is the echinoderm larva. The tornarian is the hemichordate larva. And in some species, you can't tell the difference between the 2 unless you're like, that's your expertise. If I put them on microscope slides, you'd be like, I have no idea the difference between them. They look so similar. So their morphology of their larva in particular really says, hey, these guys are pretty closely related, and the DNA suggests that same inference, if you will. Now, this is a trochophore up here. So this is a larval trochophore. So remember I talked about Lophotrochozoa and how the Lophotrocha, this is the tuft of tentacles. And then this is the skirt of cilia, this little white band that wraps around them. And that's where the name Lophotrochozoan comes from the tufts of cilia for feeding and the cilia skirt that wraps around the surface of the organism. So hopefully that gives you a little information. So basically, for now on, when we start talking about taxonomy, we're going to do kingdom Animalia, subkingdom Bilateraria, infrakingdom Deuterostomia, and then our supraphylum is Chordata. And now we're going to start talking about these 3 phyla. Okay? We're going to drop these and work our way and start talking about the groups. Whew, right? Okay, so our new superphyla, chordates and amblycarians are the two that we're interested in. And if you look at the larva, it makes a lot of sense. This is what a larva looks like of a amblycarian. It's ciliated. It almost looks like a really fancy hat that you would stick on your head, like the Pope wears. And so that's kind of what an amblycarian looks like. And the larva looks the same for a hemichordate versus an echinoderm. Look at the larva of a chordate. It looks nothing like that, right? So obviously the hemichordates look more like echinoderms than they do chordates. You can just see that in a larva, right? The larvae look completely different in a chordate than they do in an amblycarian. So notice the chordate larva has a tail, right? That's what this is a long tail. And you see that big thing stained in blue that runs all the way down the tail? That's the notochord, right? So the notochord is really important in the movement or swimming, if you will, of the larval stage. So when you look at the chordates, notice you have cephalochordata on our phylogeny. And then here are your two sister taxon called Urochordata and Vertebrata, all right, or vertebrates. They're called olfactors. There's a name that they use. I have no idea the taxonomic implications of that name, but this is a new name that they're using, which refers to the pharynx, right, or an extension of the pharynx, and the ability to smell, right? That's where olfaction comes from. And if you look at the pharynx of a vertebrate, the throat, and how it develops, and the pharynx of a urochordate, they're very similar complex pharyngeal regions. The cephalochordate's pharynx is really just a specialization of its gut. So it's less complicated, more primitive, or more ancestral. So that's why the urochordates and vertebrates are also called olfactors. And they have a larva with a tail and muscle tissue to bend it so that they swim really well. Now, I pulled a phylogenetic tree out of that paper from 2018. That's when I started teaching it different in here. There are a couple different papers that came out. This is all based on DNA. This is the DNA tree. I like to go through the names. Now, I didn't go through all of them, but you can see the names and you understanding what they mean kind of helps you remember them, right? So up here in black, these are my protostomians, right? So Lophotrochozoans and Ecdysoans, right? There you go. And they form two really nice monophyletic groups using molecular genetics. Here is Amblycaria, right? So you see this branch right here. So protostomes up here in black, and then deuterostomia, right? And then you can see the deuterostomes, they branch. We didn't talk about this group, don't worry about it. But you can see you got hemichordates, and echinoderms are really nice sister taxon, right? Nice monophyletic group that they call Amblycaria, which means to ample or walk along the floor. Then if you follow the branch, you go this way. Here are chordates, right? So here's chordata. And then over here we have cephalochordata. So here it's a more primitive or more ancestral group. And then here are our two olfactors, right? So you have olfactors and then craniata here. So the olfactors include the cyclostomata here and the gnathostomata. All right, we'll talk about these two names in a second. So these are what we're going to call our vertebrates. And then notice the sister taxon is tunicata or tunicata. These are urochordates. So again, they make a nice sister taxon. So you can see that's where those names come from. Now, cyclostomata. So stoma, I already told you about, it means mouth, right? Cyclo means round mouth. Gnatho means jawed mouth, like a movable jaw, lower jaw. So we're going to talk about these two craniates here in a second.